epithelium is giving us glands, and these glands can be classified due to certain components or the type of secretions they are having. In the beginning, we can talk about three types of glands according to their mode of secretion. That could be merocrine or apocrine, or it is holocrine. When you say merocrine, it means only the secretion is given at the cell surface. The lysosomes will fuse and they give out the secretion. There is no loss of cell membrane. That is merocrine. But sometimes there is a loss of apical cell membrane and which will then cover the secretion. It is called apocrine. Sometimes it may happen that entire cell membrane is broken, the cell organelles are broken, and entire cell become the secretion. That is called whole of the cell getting compromised. So holocrine type of secretion. Then there are other classifications, like one of that could be a serous type of gland or mucinous type of gland. Serous means they'll be giving some thin watery secretions. Mucinous is thick viscous. The serous cells are taking pinkish stain, darkly stained, as compared with the mucinous, which are lightly stained, because they have more of mucus type of secretions which take less stain. The serous glands appear more like a central spherical nucleus, cuboidal, whereas the mucinous glands, they are appearing like columnar because of the secretion. The nuclei push to the periphery, and there is a lot of cytoplasm above the nucleus appearing like columnar. Sometimes this seromucinous can be together like submandibular salivary gland. Then you can have what is called as serous demilunes. So these are all things which we want to discuss, and we can start with the first classification, which is merocrine or apocrine or holocrine. Now, as we talk about the merocrine secretion, where I'm telling that lysosomes will come to the surface and give the secretion out without no loss of cell membrane, one example is the salivary gland. Salivary gland is merocrine type of gland, and even most of the sweat glands are merocrine variety. And then you will see that when we are looking at the histology specimen, they are having the lumen, means we have to magnify this. We can see the salivary gland, the lumen, as you magnify. Since there is no loss of cell membrane, the lumen intact, the lining of the lumen is intact, means there is no break in the lumen of the ducts or glands. As we see, they are smooth appearing. You will compare it with the apocrine, then you'll find the difference. As we focus upon the apocrine, like mammary gland is a kind of apocrine sweat gland when it talks about the secretion of lipids, when you're putting lipids into the milk. So that time it becomes apocrine, means apical cell membrane is lost and is covering the secretions. And these glands, as you compare the apocrine with the merocrine, their lumen is larger as compared with the Merocrine variety. And you see here now there is loss of cell membrane which is covering the secretion, and the lumen is not smooth as you found here. Let us magnify it further. You can see the difference. The cells are losing the apical cell membrane, blebs are being formed, and these blebs then they are taken off and they will cover the secretion. Like I am telling, the lipid in the milk is given by this variety. This is apocrine type of secretion. There is some loss of cell membrane, apical cell membrane, becoming the covering of the secretion. On the other hand, if we are talking about holocrine type of glands like sebaceous gland related with the hair follicle, you'll find entire cell membrane is broken. The organelles are broken, all of them becoming the secretion. So like with the hair follicle, you can have sebum secretion. Here, if you magnify this diagram, see, this is the skin epithelium which is coming down around the hair follicle. This is hair follicle, and that is the sebaceous gland which we are calling as holocrine. We are magnifying it. This is the hair follicle with the stratified squamous epithelium of the skin, but this is the sebaceous gland. The point is, the sebaceous gland cells at the periphery may have intact cell margin, and there will be intact cell organelles like nuclei, but as they move towards the duct of the gland, they lose cell membrane, they lose their cell organelles, and they are all broken, and they are all becoming secretion like sebum to go along the hair follicle and lubricate the skin. So holocrine, whole of the cell is destroyed to become the secretion. Now this skin specimen which we are looking at, we can magnify it because it has some more details to discuss. Like you can see the hair follicle is controlled by a smooth muscle structure here, erector pylorum. Sympathetic system will work upon the erector pylorum to erect the hair. For example, fight or flight reaction. The sympathetic system will work on the erector pylorum, and there will be goosebumps, basically erection of hair. And you can also understand that along with sebaceous gland in the skin, you'll have sweat gland. So sweat gland, which is on the upper level, they are the merocrine. At the lower level, they are the apocrine. Though apocrine sweat glands are very few in number, but they can be seen in the skin. If it is deeper level, it will be apocrine. If it is a superficial level, it will be the merocrine. Actually, merocrine is a term which can be used for the sweat gland, it can be used for salivary gland, the secretion which has no loss of cell membrane, just the secretion going on. But in the sweat gland, we can specify it as acrine also. Sweat glands are two types, apocrine or acrine. And this acrine is nothing but the merocrine variety. But be careful, when we use the term merocrine, acrine as interchangeable, be careful to tell that acrine refers to only sweat gland. Sweat glands are two types, acrine and apocrine. And the merocrine term is basically applying not only to sweat gland but also to other gland. Like we have just discussed, the salivary gland is a merocrine variety of gland, but you cannot say acrine. The term acrine is applied only to sweat gland. Sweat glands are acrine and apocrine, though acrine is a type of merocrine variety only. So under merocrine, you can have several glands, and one of them is sweat gland acrine variety. And acrine term is restricted only for sweat gland, though you are using it interchangeably. So can we magnify this diagram, because we get questions upon this kind of diagrams. And as you see, this is the hair follicle. This is marker B on the erector pylorum, the smooth muscle. A is the superficially located, the acrine type of sweat gland, which can be considered as merocrine. Merocrine, acrine is same. And as you are talking about the D, it is the deeper location. So they are apocrine type of sweat gland. Now, what is the difference between the two we want to discuss? And marker C is, of course, the holocrine type of gland, which is sebaceous gland. In the periphery, the cells are intact, but towards the duct, they are broken, all the organelles are broken, and now the sebum is formed, entire cell becoming sebum itself. So what is the difference between these two types? You can see one schematic diagram here, also stratified squamous epithelium, which is coming down along the hair follicle. Along the hair follicle, you have sebaceous gland, which we just discussed, and this is the erector pylorum we were talking about. Erector pylorum can work under sympathetic system for erection of hair, the goosebump. And holocrine gland is the sebaceous gland, whole of the cells broken to form sebum. But when you are talking about sweat gland, they are mostly acrine sweat gland, and some of them could be apocrine. Now, acrine sweat glands, first of all, they give watery secretion, and they are duct open at the surface, the actual sweat which comes. So like in the palm sole, you will have a lot of acrine glands. So acrine sweat glands, they are superficial in the dermis, and they give watery secretion, and the ducts are opening on the surface, as compared with the apocrine variety. Apocrine variety, the secretions are viscous, thick and viscous, and they are rarely found sweat gland, and they are duct open into the hair follicle. So the secretions will come along with the hair follicle. And moreover, apocrine sweat glands are very few in number. Like if somebody is asking, where do you find apocrine sweat gland? You say they are limited, like in the axilla with the hair follicles, peri-nipple level with the hair follicles, perigenital, perianal. So in the perineum, you can have hair, and along with that, you have the apocrine variety of sweat gland. One more thing, you will see sweat glands which are deeper at level, and the acrine are at a social level. If you are comparing the two, you will find acrine are taking less of eosin stain, and the apocrine take deeper stain. And I'll show you the diagram again. In the previous diagram, we have compared the acrine with the apocrine. I told you that acrine gland, since there is low loss of cell membrane, the margin of the lumen is smooth. But here the margin of the lumen cannot be smooth. There will be some blebs form. So irregular margin of the lumen. And there are some other things which we can again look at. One more difference which you find between acrine sweat gland and apocrine is acrine sweat gland uses acetylcholine as the effector. It is sympathetic cholinergic system working here. But for the apocrine, it uses adrenergic system. So it is sympathetic adrenergic system. For the apocrine variety, it is the sympathetic adrenergic system. What for the acrine sweat gland, it is sympathetic cholinergic system. And as we are telling, there can be some modified apocrine sweat gland also. Question comes, name some modified apocrine sweat gland. The ceruminous gland, the wax gland in the external ear is a modified apocrine sweat gland. And even the mammary gland when you talk about the secretion of the lipid. So secretion of the lipid into the milk is done by the apocrine method. Mammary gland is also modified apocrine sweat gland, though it is to be understood that when you're talking about the protein secretion or other secretion in the milk, they'll be done by Merocrine variety. That is Merocrine. But lipid secretion is by apocrine variety. So you have taken two examples of modified sweat gland, which are apocrine variety, ceruminous gland, the wax gland, and the mammary gland, while it comes to lipid secretion. Now, as I'm telling, if you compare the two histologically, what is the difference we have seen? But let me repeat it again. If you're talking about the merocrine or acrine type of sweat gland, you find as you magnify, the lumen is intact. There is no break in the lining of the lumen. Lumen is smooth lining. This is acrine variety or merocrine variety of sweat gland. But if you compare with apocrine variety, first of all, you see the lumen has become larger. The lumen has become larger and the lining is not smooth because there will be breaks in the cell membrane. As you see here, there are blebs formed and those blebs are covering the secretions. Like we told that if we are talking about the modified apocrine sweat gland like the mammary gland, when we are talking about the lipid secretion, it was the secretion being covered by the apical cell membrane. So comparing the sweat gland also, like in the skin also, this is the acrine variety, which is at a higher level and sympathetic cholinergic, giving the watery secretion, and this is at a deeper level and it is adrenergic sympathetic system and it is giving thick viscous secretion along with the hair follicle. Apocrine variety of sweat gland and acrine variety of sweat gland, they are some histological differences seen here. And the other classification we told about the glands is that glands like parotid, salivary gland, they are mostly serous type of secretions. But if you look at the gland like under the tongue, sublingual salivary gland, it is mostly mucinous type of secretion. Whereas if you look at the middle, that is the submandibular salivary gland, it is mixed type of salivary gland, means it is not only serous but it is also mucous, seromucous, mixed salivary, and they are the one who will have serous demilunes also. Now, before you talk about serous demilune in the submandibular salivary gland, let us look at the parotid salivary gland. Why we are telling that it is serous type of gland? You will see they are serous cells and the epithelium is appearing like cuboidal. You see central spherical nucleus. Height of the cell is equal to breadth of the cell. This is the lumen here. So the epithelium, which is epithelium, the gland appear central spherical nucleus, cuboidal type of cells, and they take more of eosin, pinkish stain. Now you are comparing it with what? We are comparing serous cells with the mucinous, because mucinous cells, they take lighter stain and they are not as pinkish as the serous cells, and moreover these mucinous cells, their secretions are mucinous, thick, and the serous gland secretions are thin and watery. The mucinous cells are also identified with the fact that there is so much of collection of the secretion, the nuclei are pushed to the periphery. and there is a lot of cytoplasm above the nucleus. So it appears like columnar cells because the nuclei are pushed to the base. Now let us draw this diagram also as you are comparing the mucinous. Say this is the mucinous gland, this is the lumen. Look at the cells. These cells, you will find nuclei are at the base. So there is so much of cytoplasm of the nucleus. It appears like these cells, they are appearing like columnar cells. Now, if you compare it with the serous type, you can see for yourself the nuclei are in the center. Height is appearing equal to the breadth, so cuboidal. The point is, when you are talking about serous cells, cuboidal cells making the gland, the serous acinus, and if you're talking about mucinous, like the sublingual salivary gland, then you find that they are appearing like columnar cells. But if you are having a mix of that, like submandibular salivary gland, mixed salivary gland, what do you see? I find there are serous acini which are identified by the fact that they are darker staining compared with the mucinous which are lighter staining. Plus you see the magnified view now. Let me zoom this area to have a better view, and you will find now serous demilune here. First of all, you can see for yourself, this is the cell which is having the nucleus pushed to the periphery, a lot of secretions here. So this is basically what we have drawn here, the mucinous acinus. So this is the mucinous acinus. We are calling mucinous because the nuclei push to the periphery. And this one, as you see, is the serous acinus. This one is serous. How do you know it is serous acinus? Because central spherical nucleus, central spherical nucleus. A cap of serous gland on the mucinous gland is called serous demilune. This is what we call as serous demilune. Let me show that in the slide also. Can you see a cap of serous gland on the mucinous gland? So how do you know it is mucinous? Because look at the nuclei, look at the nuclei, look at the nuclei. They are at the periphery. And color. This is lightly stained, the mucinous acini. And if you are talking about the serous, it is darkly stained. One more. You see, this is a serous cap. This, let us keep the color code same as we are telling. This is the serous cap on the mucinous acinus. This is the serous cap on the mucinous acinus. So this is the mucinous acinus. So how do you know that this is mucinous acinus? We have found that mucinous acini, first of all, they'll be lightly stained compared with the serous, which is darkly stained. And serous, if you look at the serous cells, they have this typical spherical nucleus, but that is not in the mucinous. The secretion of the mucinous gland were pushing the nuclei to the periphery. So when you find seromucinous together, like in submandibular salivary gland, in submandibular salivary gland, you will find that serous mucinous acini are together. Then you will find this serous demilunes. So serous demilunes can be seen in the mixed type of salivary glands, that is submandibular salivary gland. One more thing, do you look at the duct here, duct of the salivary gland? When you compare the duct of the salivary gland, may it be the submandibular salivary gland, or maybe it is the duct of the mucinous salivary gland, which is obviously the sublingual salivary gland, or maybe it is the duct of the parotid salivary gland. See, we have told parotid salivary gland is mostly serous acini, this is the duct, and sublingual salivary gland is mostly mucinous, that is the duct, and submandibular is in the middle and it is mixed, it is seromucinous and it is the duct. These ducts of the salivary gland, they have striations. So this you have to learn that this is the duct. Duct will have these cuboidal cells, and these cuboidal cells, this is the lumen here. With the saliva, you will have saliva in the lumen because the salivary gland. The cells, they will have basal striations. They have basal striations. The ducts are striated ducts. Now, what is the use of having the striations, the basal striations? That is for the concentration of saliva. In the basal striations, there are some ion transport happening. So because of that ion transport happening at the basal level, there are the basal striations, and that makes the duct striated. Ducts are striated. Compare it with the pancreas. Pancreas, they do not have striated ducts, because in the pancreas the ducts do not have to concentrate the secretion. There they are just carrying the secretion. Like in the pancreas you have bicarbonate alkaline rich enzymatic secretions are there in pancreas. You don't have to actually concentrate anything. So pancreatic ducts are not striated. Why I'm telling you this is because there comes a comparison of pancreatic histology and the salivary histology. So how will you differentiate between the two? That is what I'm telling. Let me show you a diagram also. Like I'll show you the parotid salivary gland. Parotid salivary gland histology looks like pancreatic histology. So how will you differentiate the parotid salivary gland from pancreas? Because in pancreas also you have serous acini like this only. You have ducts like this only. So let me show both the slides side by side because that came as a question. In fact, this is not parotid salivary gland. This is submandibular salivary gland because you see serous acini and the mucinous acini also. But then maybe it is serous acini plus mucinous acini and it is a submandibular salivary gland. This duct, as we are showing, these ducts, they are special. These ducts, they will be having some basal striations. And why do you need those striated ducts? We need the striated ducts because we want to concentrate the saliva, and that is not seen in the duct of the pancreas. This is pancreatic duct. So pancreas duct, but obviously you cannot see the striations at this level. You need a more magnified view, then you can be seeing those striations. But the other thing is, can you see something else in the pancreas? In the pancreas, not only you have these pancreatic acini, which you may be confusing with the, parotid salivary gland, because parotid salivary gland will also have the same serous acini, but in the pancreas you have islet of Langerhans with the alpha, beta, delta cells giving you the insulin. So beta cell giving insulin or glucagon is given. So these are islet of Langerhans. Point is, how will you identify pancreas slide from a slide of parotid? Because parotid slide the serous acini and pancreatic acini, they appear the same. I'll identify by the fact that in pancreas there will be islet of Langerhans, which are obviously missing from salivary gland, number one. Number two, the ducts, they are not striated. They don't have to concentrate anything. They're just carrying some enzymes secreted by the pancreatic acini. That could be some lipase or proteases, and I just have to carry them with some alkaline rich fluid. There's no concentration going on, but that concentration of saliva which is happening in salivary gland makes the duct striated. So these are the differences which you can use to differentiate the slides of parotid salivary gland or submandibular salivary gland from the pancreatic gland, which is exocrine when you come to the serous acini because they pour the secretion into duct, and endocrine also because islet of Langerhans are endocrine. They pour the secretion into the blood vessels, some capillaries around. That is endocrine nature. So it is endocrine plus exocrine. Pancreas is endocrine plus exocrine. The salivary glands are purely exocrine type of glands.